Hi, it's my pleasure to introduce Rachel Evans. Rachel is another career development fellow who's funded by RadNet, but the other half of her job is she's actually an AZ-KCL fellow working within the translational medicine branch of AstraZeneca. So we're hoping that she can link us to the translational work at AstraZeneca. She's going to talk about today about radiation IO combination to target the tumor-associated macrophages. Rachel.
Thank you very much for having me here to talk today. So as Tony said, I'm going to be talking about radiation and IO combinations and how we target macrophages. So a little bit about my background. My background really leads me to look at the tumour microenvironments in a holistic way. So my PhD was focused on T-cell biology, and I did my PhD under Nancy Hogg at the LRI. I spent the majority of my postdoc time in Tony Ng's group, where I used a variety of different advanced imaging techniques to stratify patients and really focus on the changes in the immune microenvironment, so looking specifically at macrophage signalling pathways. Over the last couple of years, I've been based at UCL, working within a group that focuses on antibody and CAR T-cell engineering. During that time, I've also started to develop my own research niche, really focusing on non-small cell lung cancer. So as we all know, lung cancer is an area of clinical unmet need, and this disease is really very aggressive and driven by the immune system. What's interesting to me is that there's a high macrophage infiltrate in the majority of non-small cell lung cancer patients. There has been a lot of success seen in treating patients with advanced disease with different immunotherapies, and much of this has focused on the inhibition of immune checkpoint signaling pathways and the effect that this has on T-cell biology. But what we know is that immunosuppressive macrophages can interact with T-cells and can also influence the effect that T-cells are having in the immune microenvironment. So here, what you can see is macrophages interacting with CD8 T-cells in the periphery of a solid tumour, and they're actually preventing the T-cells from infiltrating further into the tumour microenvironment. And so what I want to address is whether we can reprogram pro-tumorigenic macrophages into an inflammatory and anti-tumour phenotype. So what I want to be able to do is use radiation therapy in order to do this. So we know that lung cancer patients are currently given radiation therapy as a mainstay of their treatment protocols. In recent years, clinical trials have focused on combining radiation therapy primarily with anti-PD-1 or PD-L1 therapies, so members again of the immune checkpoint signaling. And the results from this have shown success. So adding in molecules such as durvalumab with radiation therapy gives a survival advantage. However, the role that this kind of combination therapy has on macrophages is not particularly well studied. However, what we do know is that radiation really, the effects of radiation are changed throughout time. And so radiation can have different effects early on and late on. So early post-radiation, what we see is an inflammatory response that's really driven by DNA damage. But as this starts to resolve, what we can get is a chronic, what you can see here on the right in this schematic, is a chronic wound healing response. And I won't talk through all the signaling pathways. There's a number of signaling pathways that occur here, but they're primarily immunosuppressive. And what it culminates in is some very important changes in the microenvironment. So this includes remodeling of the collagen structures and the release of latent TGF-beta into its active form, and a number of different signaling pathways. And all of this really culminates in the skewing of tumour-associated macrophages towards an M2 immunosuppressive phenotype. What I'm interested in is trying to prevent this from occurring, and essentially maintaining macrophages in a more inflammatory phenotype that can essentially target the tumour. Some of the things that we'll need to consider when we think about this in the clinic will be our radiation protocols, so the type of fractionation and dose that we're using, but also the correct selection of IO agent. So not necessarily sticking to the same PD-1 and PD-L1 inhibitors that we always think about. The timing of these IO agents will also be critical. Oh, sorry. Not sure what I've done here. Apologies. Okay, sorry. Okay, sorry about that. Okay, so this project will really focus on two main questions. As I mentioned, we're going to try and redirect macrophages towards an anti-tumour phenotype, so really instigate an inflammatory response. And we also want to understand our combination protocols from a molecular level through to the whole body level. And this becomes really important when we start to think about personalising medicine in the clinic. So it's important to really take into consideration a patient's individual microenvironment. So I'll talk you through briefly the main parts of the work that we've just started. So at the moment, we're at the point where we're defining our macrophage subpopulations in lung cancer models. So we're primarily using a lung cancer model from Sergio Quesada's group. And this is similar to the model, one of the models that Rita's using. And it's derived from an inducible mouse model, which has a mutation in KRAS and P53. So Sergio's group have derived a cell line from these tumours, which can be injected into mice and ultimately leads to the rapid growth of multiple lung nodules. We're now in the process of looking at the macrophages associated with the tumours and how they change over time. And we're using immunohistochemistry and multiparameter flow cytometry. We're taking quite a broad look at some of the phenotypic and functional markers that are expressed on these macrophages, but we're focusing on molecules that we believe that may be key in their response to radiation therapy, so including PD-L1, TIM3, and TGF-beta. It's important for us that we make our work as translational as possible. And so one of the ways that we're doing this is collaborating with Rob Hines and Charlie Swanton's group, and we're using patient-derived cell lines from the TracerX study. And we're making a very simple co-culture experiments where we essentially culture with donor-derived monocytes and then start to look at the macrophage populations that come from this co-culture, again using multi-parameter flow and mass cytometry. We're now at the point where we're beginning to look at the effect of radiation therapy on macrophages and both their phenotype and function before we start adding in different immune checkpoint inhibitors, and we'll do that either concurrently or sequentially. And we're doing that with the help of Rebecca Carter at UCL. Once we've selected agents that we're interested in using and combining with radiation therapy, we'll be looking at the therapeutic distribution of these drugs within the solid tumour. And we'll be doing this by radio labeling our drugs and using whole body imaging techniques in collaboration with Tammy Kalber at UCL. So as Tony mentioned, I'm working closely with AstraZeneca, so I'm within their translational medicine team. And what this affords me is access to a number of their different patient data sets. So at the moment, I'm currently defining some of the baseline macrophage signatures that we see in lung cancer clinical trials and trying to correlate that with some of what we see in our preclinical work. As our projects start to evolve, we'll have access to some of the clinical trials in AstraZeneca where radiation has been combined with IO agents. And we'll be able to look at how some of these macrophage populations change pre and post therapy. Ultimately, what we want to be able to do is use some of the available compounds at AstraZeneca and use these in combination with radiation therapy. And this will all be guided by data that we get from the clinical trials. Of course, it will be important to understand more mechanistically how effective combination therapies are working. And so we're using an array of different imaging strategies. So initially, we'll start tracking macrophages at the whole body level, and this is going to be done in collaboration with Samantha Terry at KCL. We'll also use multiphoton microscopy of living lung sections, and this will be done with help from Leo Carlin at the Beatson Institute. But ultimately, what we're interested in is instigating an inflammatory reaction in the macrophages that are within the tumour microenvironment. And so we hope to really quantify that using a STING biosensor with help from Tony Ung's group. And we'll be able to express this biosensor in macrophages and use these in our 3D co-culture systems and use fret film imaging to be able to quantify how we're redirecting our macrophages. And so with that, I'd like to finish with an acknowledgement slide that just acknowledges all the funders, but in particular, Jan Lau, who has recently joined me at KCL to take on the majority of the wet lab work, and also Dominic Patel, who's been doing the histopathology for us at UCL. Thank you.
Thank you, Rachel. Maybe I can start. So, what is known about macrophage populations in lung cancer in response to different radiation modalities, so fractionated radiation versus, say, flash radiation, so in terms of, you know, a real chronic treatment? So
generally what happens is that you do see this initial inflammatory response, but then in terms of long-term effects of the different radiations, it's not very well described at all. In fact, there's not really been much radiation biology apart from sort of standard radiation therapy and these sort of long-term effects and the sort of general wound healing response and immunosuppressive microenvironment. But how the different radiation therapies compare is not well looked at. And so, yeah, that's sort of one of our starting points, really. So a lot of profiling.
Hi, that was a nice talk, Eric Sahai from the CRUK again. I'm just interested in your co-culture work. Are you just thinking of putting the cancer cells with the monocytes from the patients, or are you going to try to get more complex than that?
So we would like to, so at the moment it's a very, very simple setup. It's 2D and it's not even patient-derived monocytes. So there are many things that we would like to change about that. We would like to make it into a 3D co-culture. And ideally what we would want is patient-derived PBMCs. So to have sort of a more holistic view rather than just monocytes.
Okay, thanks.
I think we have some questions online as well. Eric's going to read those.
Okay, so this still sounds like my voice, but it's actually Rob Rulak. Fascinating stuff. Radiotherapy can have quite a high pneumatitis rate and can make patients quite unwell. Could you explain how you will show that the reprogrammed macrophages won't perpetuate normal tissue toxicity? Thanks.
Well, initially, I guess this is something that we need to be wary of in our mouse models. So this is, initially we will be looking exactly at that, so toxicity effects. For sure, it's going to be a real issue when we add in different molecules as to whether or not we get sort of too much of an inflammatory response. Yeah, that's something that we need to take into consideration with mouse models, I think, before we think about patients.
Thank you, Rachel. That was great. Thanks very much.
Thank you.